So students, in this lecture we are going to study the bacterial growth, culture media and the culture methods. So till now we have studied the structure of bacteria, what the bacterial cell possesses, what are the structure which are present outside the cell wall, what is the cell wall, what is the nature of cell wall, what are the different structures which are present inside the cell wall. Now we will study how the bacteria replicate, how the bacteria grow, what are the requirements for their growth, and what are the different nutritional requirements or the different culture medias which are used to grow the microorganisms. Already we know that the bacteria grow by binary fissions, resulting in exponential increase in number. That means the one cell of bacteria results in two cells, from 2 to 4, 4 to 16. Likewise, the 4 to 8, 8 to 16. Likewise, the bacteria number grows on increasing. The number of cells arising from single cell follows the 2 raised to n formula after n generation. And the generation time is the time it takes for a single cell to grow and divide. Means the time required for a cell to grow and divide into two cells is called as generation time. So here we can see this is the species A, this is the species B. Species A grows from here to here. These are the number of cells and the time. The number of cells and the time required for the growth of cells. This is how the binary fission, the bacteria divides by binary fissions. Let us take an example of this bacteria. It is like a Bacillus subtilis, a rod shaped bacteria which contains the cell wall. This is the nuclear material. This is the yellow color line is the plasma membrane. Okay. So this bacteria, the first step, the DNA or the nuclear material, it's elongated and it starts to replicate. In second step, the DNA get completely separated. They get completely replicated, and the cell wall and plasma membrane will start to divide the cell in equal parts, and the cell will start to form a septum in between of the cells. In third step, there is a complete cross wall is formed which divides the DNA into two different segments, and in fourth step there is a complete cell separations giving rise a new cells, new replica of the mother cells. This is the rapid growth of bacterial population expressed in a number. You can see here, this is the first generation which is having only one cell. The second generation, the one cell gives two cells, that is 2 raised to 1. In next generation, third generation, these two cells will give rise to fourth cell, then 4 to 8, next 16, 32 and so on. This is how the bacteria grows exponentially, and the time required to grow this bacteria from 16 cells to 32 cells, or from 4 cells to 8 cells, or from 2 cells to 4 cells, that time required to replicate the bacteria, to grow the bacteria, is called as generation time. This is the growth curve of bacteria. As you can see here, there are total 4 phases in the growth curve of bacteria. The first is initial phase, that is the lag phase. Second one is a log phase, that is also called as exponential phase. Third one is stationary phase, and fourth one is decline or death phase. So in first phase, during lag phase, the cells are recovering from a period of no growth and are making macromolecules in the preparation for the growth. That means The cells are adjusting themselves for the growth. They are taking the nutrients from the surrounding, and they are adjusting with the environment for their growth. In log phase, the cultures are growing maximum. That means there is a maximum growth of microorganisms. The cells continuously go on dividing by binary fissions, and they start to replicate themselves in number of times. In third stage, stationary phase occurs when the nutrients are depleted and wastage accumulate. That means during log phase, what are the possible culture available in the surrounding, or what are the possible nutrients available in the surrounding, these bacteria or these cells will try to acquire or accumulate the nutrients, which will decrease the amount of nutrients. At this phase, the number of bacteria or the number of cells which grow are equal to number of cell death. That's why it gives a straight line, that is stationary phase. Means growth rate is equal to death rate. And in fourth stage, the death rate is greater than growth rate, because there is no more amount of nutrients available for the growth of microorganisms. Eventually the cell will start to die, and the death rate will be more as compared to growth rate. So it will start to decrease the number of cells into final step or final phase. So as the microorganisms, they also require the nutrients or nutritional requirement, which the microorganisms or specifically the bacteria consumes, and they grow. So microorganisms or microbial nutrients and growth require certain physical and chemical parameters. So there are certain requirements or nutritional requirements that are required for the growth of microorganism. So physical parameters that are also required, which affect the growth of microorganisms, and the classification can be done based on this. There are certain chemical parameters or chemical requirements that the bacteria required for their growth, and the classification can be also done based on the chemical parameters required for the growth of bacteria. So environmental factors that are required for the growth of bacteria is the temperature, pH, osmotic pressure, and the oxygen. So different classes of bacteria require different conditions for their survival or for their growth, and the bacteria can be classified based on that. For example, temperature. Temperature is one of the important factor for the growth of microorganism. Those bacteria, some of the bacteria which can grow at minimum temperature, some of the bacteria can survive or can grow up to 80 degree or 90 degree Celsius. Some bacteria required less pH, or they can grow or survive in acidic conditions, or most of the bacteria are there which can grow or survive in alkaline conditions. Some bacteria can tolerate the extreme tonicity, whereas some bacteria are stable, or they can survive in osmotic conditions only. In case of oxygen, some bacteria can grow in presence of oxygen, some bacteria can grow in absence of oxygen. Some bacteria are there which do not require oxygen for their growth, but they can survive in presence of oxygen. So all these requirements classify the bacteria according to their requirement for the growth or for the survival. So the first environmental conditions required for the growth of microorganism is temperature. So temperature is one of the important environmental factors which decides or which affect the growth of microorganisms. Based on their growth or the nature of growth, the temperature, it is divided into three segments. The cardinal temperature is the temperature where the actual bacterial growth takes place. In that, the minimum temperature means a minimum temperature required for the growth of bacteria. Optimum temperature means this is the temperature range where the maximum growth of bacteria is possible, and the maximum temperature is, it is a maximum temperature which shows the bacterial growth, or bacteria can grow in that maximum temperature. Below minimum temperature, the bacterial growth is not possible, and above maximum temperature, bacterial growth is not possible. So, here we can see in this diagram, this is the growth rate of the bacteria and this is the temperature range. We can see here, this is the minimum, this is the optimum, and this is the maximum. In case of minimum temperature, below minimum temperature, the bacterial growth is not possible. This is the optimum temperature where the enzymatic reactions occurs at maximum possible rate. The bacterial growth is maximum in the optimum conditions, and the maximum temperature is the maximum temperature where the bacterial growth is possible. Above this maximum temperature, the protein starts to denature, which collapse the cytoplasmic membrane, which results in thermal lysis of the bacteria. That means the bacteria will starts to die because of the denaturation of proteins. So, this is the actual temperature and this is the cardinal temperature right from this to this, where the bacterial growth is possible at different speeds or different rates. From minimum to optimum temperature, the enzymatic reactions within the bacteria occurs at an increasing order. At the optimum temperature, the enzymatic reactions is at maximum possible rate, and after the optimum temperature, again it will starts to decreasing, and this is the maximum temperature where the bacterial growth is possible. So, as already discussed, the minimum temperature is the temperature below which the growth ceased, or the lowest temperature at which microorganisms will grow. Below this temperature, no growth is possible at all. Optimum temperature is temperature at which its growth is fastest. The bacteria grow at maximum possible rate, and the maximum temperature is temperature above which growth is stopped, and it is the highest temperature at which microbial growth will occur. Already discussed, above the maximum temperature, the protein denaturation starts, the membrane starts to lyse, and the bacteria dies. So, based on the requirement for the temperature, the bacteria are classified into different classes. So, this is the classification of microorganism by the temperature requirement. So, bacteria are broadly classified into different classes as per the temperature requirement. First one is psychrophiles, means the bacteria which can grow right from 0 to 10 degrees Celsius, where the 4 degree is the optimum temperature for their growth. Then the mesophiles, it starts from 8 degrees Celsius and extended towards the 45 degrees Celsius, and 37 to 39 degrees Celsius is the optimum temperature for the growth. Then the thermophilic bacteria, the minimum temperature for thermophiles is 40 degrees Celsius and the maximum temperature is 70 degrees Celsius, where 60 degrees Celsius is the optimum temperature required for their growth. Hyperthermophiles, they can grow, minimum temperature is 65 degrees Celsius, maximum temperature is 95 to 98 degrees Celsius, and the optimum temperature is 88 degrees Celsius. And the hyperthermophiles are the bacteria whose minimum temperature is 90 degree, maximum temperature is 110 degree, and the optimum temperature is 106 degrees Celsius. So, this is how the bacteria can be classified based on the requirement of temperature. As here we can see, the hyperthermophiles here can grow up to 110 degrees Celsius. That is why if we want to sterilize anything, it should have a sterilization temperature more than 110 degrees Celsius. The common example of psychrophiles is Polyromonas vacuolata, mesophiles Escherichia coli or E. coli, the thermophiles Bacillus stereothermophilus, It is the bacteria that is required. It is a biological indicator to check the sterility or the to check the to validate the autoclave. Then hypothermophiles, thermococcus sealer, and hyperthermophiles, pyrobolus fumari. So these are the bacteria can be classified according to the temperature required for the growth. So as already discussed, the mesophiles, the temperature range starts from 20 to 45 degrees Celsius, found in warm blooded animals, aquatic environments in the temperate and tropical latitude. Psychrophiles 0 to 20 degrees Celsius. The cold temperature is optimum temperature required for their growth, and the most extreme representative inhabit permanently cold environment. Then the thermophiles already discussed in previous slides. These are some of the examples of microorganisms or the bacteria whose minimum, optimum and maximum temperature is given in this chart. So you can see here the Escherichia coli, the minimum temperature is 10 degrees Celsius, optimum temperature 37 to 38 degrees Celsius, and maximum temperature is 45 degrees Celsius. Likewise, Pseudomonas, Bacillus, Psychrophilus, you can see the minus 10 degrees Celsius is the minimum temperature. Optimum temperature 23 to 24 degrees Celsius, maximum 30 degrees Celsius. The next factor that affect the growth of microorganism is the pH. So pH measures the H plus ions. Each organism has pH range and pH optimums. The bacteria which can grow in presence of acid or which can survive in presence of acids are called as acidophiles, where the bacteria which can grow or which can survive in higher pH are called as alkalophiles. The optimum pH for acidophiles is 1 to 4, whereas the optimum pH for alkalophiles is 8.5 to 11. The lactic acid bacteria having pH 4 to 7. The Thiobacillus thioxidans as having pH range 2.2 to 2.8. Fungi can grow from pH 4 to 6. So internal pH can be regulated by buffers and near neutral adjusted with ion pumps. Human blood and tissue has pH 7.2 plus minus 0.2. So apart from that, the third factor which is required for the growth of microorganism is osmotic pressure or the osmotic conditions. The osmotic pressure depends on the surrounding solute concentration and water availability. The water availability is generally expressed in physical terms such as water activity. As we know that the osmotic pressure or the osmotic conditions has a huge impact on the bacterial cells or the any type of cells. We know that if the cells are exposed to hypertonic solutions, the cells will start to shrink, and if the cells are exposed to hypotonic solutions, then it will swell. Whatever the content is there, it will start to swell and the bacteria will burst or the cell will burst. Similarly, the osmotic pressure or osmotic condition also has impact on the survival of bacterial cells. So this is the osmotic effect and water activity on the microorganisms. So organisms which thrive at higher solute concentrations are called as osmophiles, means they love the higher concentration of solute. So microorganisms which tolerate higher concentration of solute are called as osmotolerant, means they can tolerate, they will not grow in presence of higher concentration of solute, but they can tolerate that higher solute concentration, means they will not die. They are called as osmotolerant. Organisms which grow in high salt called as halophiles, which tolerate high salt concentration can be called as halotolerant. So, next till now we have completed the physical parameters that are required for the growth of microorganisms or the physical factors that affect the growth of bacteria. Now, we will see the chemical parameters or the chemical factors which affect the growth of bacteria. So, why the nutrition is important? The hundred of chemical compounds present inside a living cells and are formed by nutrients. Already we know that each and every living things require energy for their survival, and the energy can be obtained from the nutrients. Similarly, bacteria also require nutrients for their growth, for their survival. So, these nutrients can be either macronutrients, means elements that are required in fairly large amount, and the micronutrients means metals or organic compounds that are required in very small amounts, as the name suggests, micronutrients. So, carbon 50% of dry weight and the nitrogen 12% of dry weight is required. So, autotrophs are able to build all their cellular organic molecules from the carbon dioxide. Nitrogen mainly incorporated in proteins and nucleic acids. Most of the bacteria can use ammonia and can also use NO3 minus. The nitrogen fixers can utilize the atmospheric nitrogen N2 for their growth. The microbial growth requires source of carbon for basic structure, source of cellular energy ATP or related compounds to drive metabolic reactions, source of high energy electrons, reducing power typically formed in NAD or NADPH. So, apart from carbon and nitrogen, these are other macronutrients which are required in fairly large amount for the growth of bacteria, that is phosphate, sulfur, potassium, magnesium, calcium, sodium or iron. Iron plays a major role in cellular respiration, being a key component of this cytochrome and iron sulfur protein involved in electron transport. The iron binding agents that the cell produce to obtain iron from various insoluble minerals. So, these are some of the micronutrients needed by living organisms. For example, boron, the function of boron is present in autoinducer for quorum sensing in bacteria, also found in some polycatides antibiotic, and chromium, cobalt, copper used for the respiration, cytochrome C oxidase, biosynthesis likewise. And these are some of the micronutrients needed very little amount, but critical to the cell functions: nickel, selenium, tungsten, zinc, etc. Then some of the growth factors like organic compounds required in very small amount and then only by some cells. So, these are some of the vitamins and their functions for the growth of cells. Then the another requirement for the growth of bacteria is the oxygen, and most of the bacteria are classified based on the requirement of oxygen for their growth. So, we can see here these are the classification of bacteria here. The obligate aerobes or strict aerobes are the aerobic bacteria which require oxygen for their growth, and they compulsorily required oxygen for their growth. In absence of oxygen, they cannot grow or cannot survive. Then obligate anaerobes or strict anaerobes means these are the bacteria which do not require oxygen for their growth, or they cannot survive in presence of oxygen. Then facultative anaerobes grow better in oxygens, means these are the microorganisms which can grow in presence of oxygen, but they can be survived in absence of oxygen also. Aerotolerant means these are the organisms which do not care about oxygens, means they are better grow in absence of oxygens, but if it is present they can survive. And the micro aerophiles require low level of oxygen. As explained, aerobes, obligate aerobes, they require oxygen to grow. Facultative can live with or without oxygen, but grow better in presence of oxygen. Microaerophiles that require reduced level of oxygen, whereas anaerobes, aerotolerant anaerobes can tolerate oxygen but grow better without oxygen, and obligate means they do not require oxygen at all. So these are the oxygen relationship of microorganisms, obligate, facultative, microaerophiles. These are some of the examples of bacteria which require oxygen, which may require oxygen, and which require very less amount of oxygen. In this case, the bacteria can grow better in absence of oxygen, but can be survived in presence of oxygen also. And this type of bacteria, they do not require oxygen for their growth.